the Dementia Researcher podcast, talking careers, research, conference highlights, and so much more.
So we're back with part 2, recording on location from Lisbon in Portugal to share highlights from the last 3 days of this year's ADPD conference. One of the biggest conferences in the calendar, with over 4,000 delegates sharing groundbreaking science. I'm Dr Emily Maguire, and I'm a postdoc working at Cardiff University within the lab of Professor Julie Williams as part of the Dementia Research Institute. So my work is generating iPSC models of late onset Alzheimer's disease based on the genetic risk for Alzheimer's disease. Joining me to share their ADPD conference highlights are Dr Heather Snyder, Dr Timothy Siegert, and last but not least, Dr Daniel Erskine. Okay, so hi everyone.
Hi.
Hello.
Thanks for having us.
Hello. Okay, so we're going to start with a round of introductions, and also I'm going to give you a chance to talk about any work that you have or will be presenting this week. So first, Heather, do you want to go first?
Yeah, absolutely. Happy. Thanks so much for having me, and looking forward to the conversation with all of you today. I'm Heather Snyder. I'm one of the scientists on staff at the Alzheimer's Association. And in my role at the Alzheimer's Association, I oversee all of our funding portfolios. So today, the association has active and currently committed over $405 million at work in over 56 countries, more than 1,100 projects. So while I haven't presented at ADPD, it's been really exciting to see so many of our funded researchers presenting their work, the latest findings, and some of them have been presented by the researcher themselves, and some of them have been presented by their mentor and their PI. So I look forward when we talk a little bit in more detail about some of the highlights and the things that we've seen to talk about some of that work.
Awesome. It must feel really good to see some of the work that you've funded come to life.
It is, and we're a science-based patient advocacy organization, and so at the end of the day, how the science translates ultimately to our understanding of the diseases that cause dementia and ultimately translates to helping those that are affected, that's really what the work's all about. I think that's why we're all here. That's why so many people are here at this conference as well, and telling that story, and then telling that story as we go forward, ultimately to improve diagnosis, early detection, greater understanding of the disease, new paths of therapy, better care overall across the entire continuum for all of these diseases. That's the key.
Yeah, certainly. I don't know what us academics would do without people like you. So thank you.
It's an ecosystem we're all in.
Yeah. Okay. So Timothy.
Great. Yeah, and it's not just academics that get support. So we have Alzheimer's Association support to my co-founder Steve Strittmatter at Yale for part of our program, where we have clinical results that we're presenting in just a few minutes. So save some of the best for last, I suppose. Yeah. So my name is Timothy Siegert. I'm a co-founder and chief operating officer of Allyx Therapeutics. We're a small biotech company based in New Haven, Connecticut, spun out of Yale University with my co-founder Steve Strittmatter, who's the chair of neuroscience at Yale, and we're really focused on understanding and drugging the process by which A-beta oligomers interact with synapses and drive synaptic dysfunction and loss as a disease-modifying therapy for Alzheimer's. And there's a lot of support that that same type of pathway could be activated in Parkinson's disease, with alpha-synuclein aggregates being the neurotoxic species, and we have funding support from the Fox Foundation. So we've been advantageously benefiting from a lot of these different grant funding agencies, equity funding to help drive some science forward into the clinic and give some hope to patients.
Well, thank you. It's always nice to hear from someone who works in industry, having always worked in universities. And good luck later. We'll all be rooting for you.
Thank you.
Okay, so Dan, I know you've been on the podcast before, but please introduce yourself.
Yeah, hi. So I'm Daniel Erskine. I'm an Alzheimer's Research UK Senior Fellow, and I'm also a Senior Lecturer in Neurodegenerative Pathology at Newcastle University in the UK. Primary interest in Lewy body diseases, particularly Lewy body dementia. I gave a talk this morning unfortunately time really does escape at conferences, especially ones that I would say are, forgive me, but American style conferences like this one that start really early and finish really late. So time really does fly. So I gave a talk earlier about some of our post-mortem findings from both Lewy body dementia post-mortem brains, but also the brains of children with lysosomal storage disorders that have pathological overlaps with these, inverted commas, age-associated diseases. So yeah, I'm really looking forward to chewing down on everything we've been looking at over the last few days.
Nice. I hope your talk went well. You got some interesting feedback, did you?
Yeah, interesting discussions. Yeah, very much so.
Nice, perfect conference experience then.
Yeah, I think this is the value. It's something that you really can't recapitulate online. I mean, the science, to some extent, I don't think always happens in the lecture halls. It happens in the coffee shops, around the posters.
Definitely.
It's the chats and discussion, it's the collaborative nature of science. Very much so.
Yeah, it helps us put our own work into perspective and also think about it in different ways.
Absolutely. I mean, if you even look around this table, this sums up the collaboration that these conferences bring together. It's not just academics. I talk to academics all the time. I don't talk to people who work on, oligomer-based therapies, which is very relevant to my work, or science-based patient advocacy groups. It's the power of conferences that brings us all together.
I think I've done more on the stairs and had more conversations on the stairs going up and down and in that long hallway than I completely agree with you.
And it's made the forums really informative too, right? Where you have different people from across academia and industry or philanthropy talking about, clinical development and Alzheimer's disease, that a lot of these people are siloed but still have some similar and shared problems that they're trying to address.
And I also feel like it takes a variety of different kind of people from different kind of jobs and industries in order to actually bring things to the clinic overall, like rather than just one type of scientist. So, okay, we're done with the introduction. So next, let's get on to the conference highlights. So just like with part one of the show, we have a pretty simple format. So essentially, we're going to go around 3 or 4 times, and I'm going to ask you to talk about your best bits from the conference and the talks or posters that you've enjoyed the most and why. And basically, it'd be great if you could just give a summary of them and also talk about the main takeaways. So in the first part of this 2-part special, we covered Tuesday to Thursday morning. So now we're going to talk about the talks and posters from Thursday lunchtime onwards. So Heather, can you please share your first highlight?
Yeah, no, I mean, there's been so much to discuss. I think we were chatting a little bit before we went live here, and just there's been a lot of different topics and a lot of different threads of research and how it's come together. But one of the talks that I know many people from our team at the association really enjoyed was Dr. Bill Jagust, who from Berkeley, who talked about cognitive successful aging. So how do we think about successful cognitive aging? We talk a lot about these diseases that cause dementia and what is going wrong or what is changing in the brain during these diseases, but we don't talk as much about what is happening in those individuals that are successfully aging without cognitive decline. And so Bill talked a lot about some work from one of his postdocs, Dr. Stefania Pizzoli, who happens to be an Alzheimer's Association research fellow. So it was really great to see her work on that main stage as well. As we understand more about that underlying biology, as we understand more about the specific brain regions that might be at play and where there might be perhaps a greater connectivity or regional differences in the brains between people that are successfully cognitively aging versus those that may not have that same resilience, that can give us a lot of insights as we think about what are those interventions, what could be some of those strategies to target that biology maybe even earlier, so that we can strengthen that in individuals to help really in that successful aging and maintain the integrity of cognition.
Yeah, totally. I feel like it was indeed one of the main themes of this conference, the kind of resilience aspect. So what's happening in people who are kind of aging in a normal or cognitively healthy way. Yeah, it's very.
And what does that look like, particularly in the underlying biology? So I think, well, I'll talk about that next. I'm not going to give away my next highlight.
No, one highlight at a time. Okay. Tim, would you like to share one of your highlights?
Yeah, sure. So, at Allyx, we're wrapping up our phase 1B study now, and I'm presenting that. So I spent a lot of time thinking about phase 2, and I think one of the exciting things over the past, probably year, 2 years, once we've had monoclonal antibodies start showing they're successful, has been the biomarker tools that are available to measure response to drug treatments. And so I thought a really phenomenal tour de force discussion on p-tau biomarkers from Kaj Blenau yesterday was really fantastic, because one of my favorite questions to ask people, right, is, okay, we know we can remove amyloid from the brain. We know p-tau217 and p-tau181 go down, but what does that actually measure, right? And there's a lot of hand waving that says, you're measuring and that's related and a biomarker of amyloid removal. But tau is an amyloid, right? And there's an interface between those. So Dr. Blennow did a phenomenal job of like a tour de force historical assessment of p-taus and how they move not just in Alzheimer's disease, but in Creutzfeldt-Jakob's disease, which is implicated in prion protein signaling and misfolding. Across these neurodegenerative diseases, you see p-tau come up, and you see different species of p-tau come up. And so the question is, if you see the same p-tau move in a disease that doesn't really have any amyloid pathology, what does that mean? And how can you use those tools in a clinical trial to see if your drug that's targeting synapses might be having a disease-modifying effect? So I thought it was really phenomenal trying to dig down deeper into these questions for beyond just these pathologies defining disease and disease stage. What actually are they measuring in terms of response to therapies?
Well, that sounds really awesome. I didn't see that one, but I'm really glad that they have them recorded and we can watch them later, because it seems like it was definitely one to watch.
Yeah, I mean, you could give a 25-minute talk on it. If you read all the literature, it'd probably take you like 4 years to get through all of those biomarker studies.
I'm surprised you got it in 25 minutes even.
Well, it was a lot of look at this paper, look at this paper, look at this paper, and some highlights.
Well, thank you. And Dan, do you have?
Actually fits in very nicely with what you were just saying. I do think the theme of resilience has been such an interesting point. I caught a really excellent talk. Again, my perception of time is slightly altered. I'm pretty sure it was yesterday. There's a guy called Tyne Lequez. Really interesting post-mortem study that kind of adopted an almost contrary approach to what most post-mortem studies did, or what they do. He essentially looked at the slopes of cognitive decline in patients that were assessed sequentially over time, and looked at the numbers of neurons and different types of neurons, and also neurodegenerative pathologies, and basically looked for which of those associated with preserved cognition. In other words, a less steep slope of decline. Super interesting. So, for example, in Alzheimer's disease, they'd observed that layer 2-3 pyramidal neurons were associated with reserve, with a less severe decline, all other things controlled. And they also noted then with Lewy body disease, which I found very surprising, they found a microglial cluster was decreased in those. Now that's interesting to me because I look at a lot of brains, and inflammation is not something, it's something people who do models talk about a lot in LBD. People who look at brain tissue don't really so much, because you don't see obvious evidence of gliosis in the same way that you would in Alzheimer's disease or multiple sclerosis. So it was very interesting to see that microglia that we don't normally associate with LBD in the same way we would Alzheimer's disease seem to be decreased, or seem to be of lower abundance in those that appear to have preserved cognition for a longer period of time. And I just love this approach, because it really is, instead of looking for disease associations, looking for health associations, which I really liked.
So I just have to say, that is incredibly cool. It'd be really interesting if they went the next step and then looked at some of the underlying genetics to say, is there a linkage, or is there anything that is perhaps informing the immune response, or how the immune response in those individuals, is just a fascinating across all of the trajectory that they're doing.
I mean, I totally agree, and I think the immune system, I always say that there's two different types of people that work in the field. There's neuroscientists and there's immunologists, because immunology is just such a complex and difficult field. But from what I understand, so I'm prefacing what I'm saying with that in case I say something that's silly, but as far as I understand that things like the history of immune challenge, for example, can play such an important role in mediating the future immune response. So, I mean, obviously, as we come out of a global pandemic where lots of people have a very stimulated immune system, it'll be interesting to see, what risk factors emerge from this. And again, that was something that was covered in some other talks, was the COVID-19 link.
There was one right before we came in here.
There was, yes. So yeah, very interesting.
Okay, so should we do some more highlights and?
Yeah, I mean, maybe even just building off of some of this theme, and you talked a little bit about some of the p-tau work and where that is. There was a great presentation this morning from Nicholas Ashton, who is currently in Europe, but is in the process as he ended his talk and moving to Banner, and he's recruiting, I think, for people in his lab. But he did a great talk in terms of thinking about some of the other contributing factors. So as we make these measures, how do comorbid conditions perhaps influence the measure and the assay, and what does that look like? And so he was looking at some of the things around kidney function and some of that measure, and really showed that we talk about this as being, and I've heard this in talks about being this incredibly complex, or additive in terms of the complexity of how we interpret, but he showed this, what I thought was this amazing graph, which showed across all, it had to be 50, 60, 70 different measures, that the influence that kidney function has, and tau was kind of right in the middle. It wasn't really one way or the other, it was somewhat in the middle in terms of its influence, and that just really was striking to me, and that yes, we have to take these considerations into account, but the influence or the understanding we have of these biomarkers really comes into play with the clinical symptoms and the bigger picture of the disease, depending on where someone is. But as we move earlier and earlier in that as well, taking this into account and really understanding it, and the importance that real-world data will have on that understanding is paramount. We're not necessarily going to get this from cohort data. We need to have, it's going to be all kinds. And we were talking a little bit about that in terms of the multidisciplinary approaches, but needing cohort data, needing community samples, and then tying that in with real world data that allows us to actually understand how these tools are really acting when we're out in the community and part of clinical care.
Yeah, well, I think maybe to even touch on that, there's a talk that Roche gave on one of the gantenerumab studies that they ended early because the primary one failed. And they went through evaluating what their screening structure was like. And now that we have these biomarker tools and plasma biomarker tools for screening with amyloid beta or p-tau, and you want to use that as part of your patient selection criteria, what's your screen fail rate? And what they didn't mention was anything about whether they had any kidney function data on those people. And I would say, if you're running a clinical trial, right, particularly with a small molecule, you want to avoid people who have major kidney failure or dysfunction. So maybe not quite as big of a deal early stage, but in a real world setting for patient diagnosis, to make it easier to find patients, screen them, and even make sure that the drugs are working, things like that are really important.
It's good that we now have more like machine learning tools and things like that, so that we can incorporate more and more of this data in as well when we're doing things like assessing patients and that, because it's clear that it's more than just so much more than just about the brain when we're talking about these neurodegenerative diseases, right?
Yeah, the brain controls everything. So it's actually, it doesn't just stop here. We hear so much about the vascular contributions and all of those pieces as well, and how they all are intersecting with one another. The immune system, as we were just talking a little bit about.
Yeah, and I think a lot of these pathological proteins as well, there is evidence that they, are accumulated in other parts of the body as well, especially the case for alpha-synuclein. I know that there's almost a bit of a race on to find, sample different parts of the body to see if they can be used as less invasive biomarkers than lumbar puncture, which, clinicians in the United States are very happy to use. UK clinicians.
I'm not sure I'd say that.
Oh, really? That's a perception that we're always led to believe that is something that's done a lot more in the United States than in the UK.
Done a lot more in Europe, so particularly in Sweden and the Nordic countries, there's a really strong, and in fact over imaging. In the United States, it's somewhat mixed, and part of it is because PET imaging but PET imaging is not widely available, and we've actually funded some work out of Washington University, St. Louis, to look at really across the Alzheimer's disease research network centers in the United States about what some of those perceptions is. And there's a discomfort. It's not necessarily something that clinicians are doing regularly necessarily. There is a perception of side effects and others, but where people are comfortable, they're definitely comfortable. And so it's somewhat mixed in that. But I think in research studies, we do see that people are often somewhat comfortable doing that, and it is a decision between that or PET and other things. But yeah, it's definitely in the Nordic or the northern part of Europe, it is much more widely accepted, and I think EMA was really the first to come out with some guidance in that as well, much before the US. But there are now FDA approved tests in the US and other things, so there is an accessibility. It is in some places more accessible than PET might be, because there's not a PET imager, for instance, or a PET scanner, for instance.
Interesting. I did not know that.
Yeah, it's a challenge when thinking about clinical trial design in terms of site selection. because I think it is very site dependent. Who runs the site? Is it someone with a heavy neuroscience background or a neurology background or psychiatry background? I think.
Well, the comfort with doing the actual test. I mean, and that's how, do you have the resources? Do you have the facility to do it and do it in a way that is safe and right for patients? So.
I guess this is another advantage of conferences, is that it brings researchers together from around the world as well, so we can talk about how things might be different in different parts of the world.
Absolutely, yeah, and it's interesting, of course, learning from what works and what does not work elsewhere. Yeah, definitely.
I feel like it's Tim's turn.
Yeah, sure. So, another one I thought was really interesting on Thursday afternoon. So we think a lot about how amyloid beta oligomers bind to cellular prion protein. But there was a really great talk, and we think a lot about the overlap between different neurodegenerative diseases, so Parkinson's disease, but even potentially like Creutzfeldt-Jakob's diseases or infectious prion diseases. And John Collins gave a really good talk following up his recent paper on iatrogenic Alzheimer's disease, so acquired Alzheimer's disease pathology from human growth hormone samples that were in the bank, where patients were back in, I think, the 70s, were receiving human growth hormone derived from human pituitary glands. And not only is there, instances of infectious CJD from those samples, there now seems to be some evidence of infectious Alzheimer's disease from those samples. So really interesting to think. I think sometimes we're very siloed in how we think about Alzheimer's and then Parkinson's and then Creutzfeldt-Jakob's disease or frontotemporal dementia. And what I'm really interested in is, are there overlapping mechanisms within those? Not to say that Alzheimer's is infectious or acquired through routine medical care, right? That's not the case. But just to think about, how these pathologies may all start from different types of sources, right? You can get familial disease, you can get, you wouldn't really get acquired Alzheimer's disease, but it's potentially possible, right? You can, inject amyloid beta into the brain of a mice and they have neurodegeneration, same with alpha-synuclein. So it's just like an interesting thing about, are there overlapping therapeutic targets that you can go after where they might share pathologies or share mechanisms of not just spreading, but also of neurodegeneration?
We had actually worked very closely with the Alzheimer's Association, Michael J. Fox Foundation, Weston Brain Institute, and Alzheimer's Research UK several years ago on doing a program called Biomarkers Across Neurodegenerative Diseases, with that in mind, of that there are some commonalities and where things might diverge or difference, or you might have the same thing that you're testing or you're looking at from a biomarker perspective, but it may go to the right in one neurodegenerative disease and to the left in the other. Well, that's my right, that's my left, but either way. They might diverge, let's go that way, in the different diseases. And I think that gets to, some like, can you measure like microglia? Is there a particular marker? This goes back to what we were talking about before, that perhaps in one disease it will be elevated, in another disease it would be lowered, from a cognitively unimpaired individual. And so does that give you some indication, is that direction that people may go as they progress from those initial changes or from those, and that commonality, there is some, but then you also see some differences in terms of the networks of where and how the brain networks and the communication networks are overlapping among those diseases as well. So it raises so much of those questions, but there's also a lot of learnings that you can take from one to another in terms of some of the measures of CSF, beta amyloid and CSF, and what some of the considerations are for the pre-analytical processes to alpha-synuclein with some of the pre-analytical processes. And there's a lot of synergy between some of those that are overlapping as well. So it's, again, I think one of the strengths of this meeting that brings those two spaces together and in a really unique way. We've seen that in presentations. You'll have a talk on, a very focused Alzheimer's disease-related talk. Your next talk is very specific to Parkinson's disease and some of the neuropathology related. And then your next talk might be on Lewy body dementia or on fronto-temporal dementia or on something more general in neurodegeneration. And that type of intersection allows us to have some of those conversations.
Heather, I think you're arguing for a title change of the conference, beyond ADPD and beyond, maybe, to go after, there is panel discussion on ALS and frontotemporal dementia, and bringing these people together is great.
I think it's such a good point as well, about the sort of commonalities, and I think that is something this conference does really well. But I do think there's a growing consensus in the field that there are so many commonalities. And mixed pathologies is the norm rather than the exception. The idea that we, as you mentioned, overlaps between these conditions, between alpha-synuclein, Lewy body dementia, almost all of these cases have some degree of tau and Alzheimer and amyloid beta pathology. Nearly 50% of Alzheimer's cases have Lewy body pathology. Probably most of them have some degree of inflammation. Vascular changes are almost invariable, in these cases. And even genetic risk factors hugely overlap. So, APO is a risk factor for Alzheimer's and Lewy body dementia.
And cardiovascular disease.
And cardiovascular disease, of course. And of course, vascular Alzheimer's disease, most vascular risk factors are risk factors for Alzheimer's disease as well. So many commonalities, but even to dial down to the biological level, they're almost invariably associated with the accumulation of highly expressed tissue proteins. That in itself suggests commonalities because, why is the unfolded protein response not dealing with this? Why is it not being integrated through the proteasome and or the autophagy lysosomal system? It seems like there is a lot going wrong, and it seems to involve fairly similar things. And I think that is something we can potentially therapeutically exploit someday.
There's a lot going wrong, and there's a lot going wrong at different times of disease progression too, which kind of make it, a three-dimensional question problem instead of just a two-dimensional question problem.
Absolutely, but I think it's important that we embrace the fact that it's a three-dimensional question problem and not just focus on the sort of easier bits. And I think that's something that's really emerging from this meeting. I wasn't actually going to use this talk, but it feels like a good opportunity to give my example, and I'll do a shameless punt for my colleague and former supervisor, Johannes Adams, who gave a talk on mixed pathologies during here. And this is something he's really led in. And my other colleagues at Newcastle, like Lauren Walker, have really played a prominent role in trying to understand, mixed pathologies across the spectrum of neurodegenerative diseases. So yeah, this is something that I think is increasingly important.
Well, maybe I'd go, three-dimensional. I'd actually maybe add that fourth dimension back to what we were talking about before when we add in that layer of resilience. So the three dimensions might be in terms of the neurodegeneration and the progression, but that fourth dimension is the resilience component and where that figures in across the board. And I'm just gonna skip into my next highlight quickly with that because it seems like just as you were saying, it seems like the right part of the conversation. I just heard a really nice talk from one of the postdocs in Theric Pascal's lab at University of Pittsburgh, where they actually took the revised staging and diagnostic criteria that is still in draft form and is in review at this point in time from an Alzheimer's Association work group, and then they looked at a population of about 160 individuals to say where did people fit across their clinical and biological staging. And it was really interesting. They had fairly high concordance within what you would expect based on the criteria. But then there was also the discussion around where resilience fits in and how we think about that. Comorbidities are also going to be part of that, and that will be part of the, as you go and as you expand, where you think about where it's vascular, where do some of these immune markers, et cetera, as we understand more and more about them. But that idea of resilience comes into play even as we think about that diagnostic staging and biological staging. Is when people have higher resilience, they may fall into maybe outside of that particular path. They may have a higher biological staging, but a lower clinical staging because of that resilience. They're able to withstand more of the biological insult of the disease-related changes that they're experiencing. When individuals have lower resilience, they may not. This is, of course, it was in only 160 people, which were across the 16 groups, so relatively small N, but it does kind of open up the door to us to be looking at those kinds of questions. And then as we think about some of the mixed pathologies, as we think about some of those other measures, other biomarker measures, other p-tau measures, other vascular measures, and beyond, how we can actually get to that point of being very specific for the individual based on where they are in their diagnosis and what their anticipated progression might be and what their treatment path might be as well.
I totally agree, and I think that this really does speak to personalized medicine. To this idea that every person's on their own journey. There are a lot of similarities, but there are a lot of sort of nuances as well across individuals or between individuals. And I totally agree. And I think, but this starts with, we target the individual bits first, like for example, amyloid oligomers, and then we work up from there, and then and work out and I guess ultimately become better at targeting each of these individual pathologies, and it becomes a composite treatment that's tailored to the individual. It's an exciting thought.
That's what we do in oncology. That's what we do in cardiovascular disease. There's no reason. These are complex diseases of a very complex organ. There's no reason that this wouldn't be the path that we would go. think it's exciting. We've had our firsts, but we have work to do in terms of getting to our seconds and our thirds and all of those pieces together, not just for therapeutics, but diagnostics too.
Well, maybe I'll pick up there, because I thought yesterday there was a fantastic forum discussion led by Jeff Cummings and Howard Phillott, just upon like innovation in Alzheimer's disease and how do you get new therapies through the clinical pipeline, right? So now that we know there's a finish line with the approval of lecanemab, right? And we have something that could potentially be part of a combination therapy one day. How do you increase the number of therapies that are tested in phase 2, right? It's a really expensive undertaking. And then how can we get consensus around, what's a meaningful phase 2 study response look like, so that you can get interest from, investors or pharma to show that you're having that potential to have a disease-modifying therapy. And that goes beyond just biomarkers, which are a great tool, but I think biomarkers alone are a challenge. And how do you look at, clinical endpoints of the disease or composites and think about how you're creative in terms of starting to build that deeper phase 2 pipeline, so we have more opportunities and shots on goal to target different types of things, whether it's inflammation or neuroprotection, or different ways to target tau or amyloid beta, so that we can start diversifying our approaches to put more shots on goal for patients. Because I think if you just barrel all in on anti-amyloid, right, you're going to get one answer. But if you can diversify, you really can get multiple answers at the same time, right? And you can only do so much preclinically that you just have to run the human experiment and find out.
Well, it's a complex disease. It's not one biology. I think we've talked about that here, right? There's multiple biologies going on, and we need to be addressing those biologies as we think about interventions and treatments, for sure.
I think actually that sort of, I suppose, leads me on to another highlight of mine. It was a highlight of mine because, I should say it was the Lundbeck phase 2 results for their MSA anti-alpha-synuclein therapy. So this was a highlight for me because it's always nice to hear data from clinical trials, especially that seems to be quite positive. I think what it really hammers home to me, and it is directly related to what you've just said, which is understanding what is clinically meaningful. And it's so difficult. I am not a clinician. I sit in and I see differences between groups, and I think to myself, what is clinically meaningful to the patient? I know this is a very difficult thing to define, and I know it's the subject of many, many debates, but it is something that I think requires some degree of clarity just to help us interpret these results when we see them. Because it does come down, obviously biomarker improvement is such a big thing. Imaging, which I guess is a biomarker, but understanding the clinical scales, the clinical instruments, and how changes in them reflect real life measures, I think is something that really come out of that as being important.
We hear from patients that what's clinically meaningful is that they want to keep doing what they want to do longer. And that may be very different for one person or another. But it's really in that earlier stage of when they want to be able to be, maintain independence in some of those other, behaviors and functions. And that, how do you translate that to a test, to your point, is part of the challenge, and measuring things that are meaningful to at the individual level, but on a population level or in a trial population.
Maybe that's the issue. It's the test that's the problem, rather than its interpretation.
Well, I think the clinical outcomes are, not the most well developed, right? They're the tools we have at the time. But I think John Morris presented yesterday a little bit about clinical meaningfulness. Just if you get down into the weeds of what these scores actually mean in terms of if you can delay people by 3 years to the phase where they'd go from like stage 2 to stage 3, and what that means for like even just self-care and self-grooming. Can people cook for themselves their own meals? Can they take care of themselves and wash and use the bathroom? Like that's where things get really meaningful. And it's breaking the score apart from like deeper down. What do those measurements actually mean within these composites, right? A sum of boxes, right? You just think is, a tally, but what do those individual pieces mean? And what does it mean for when we now have therapeutics and when we're developing more, for how long we could delay those steps?
Well, I think, I don't know how many people in the audience yesterday, because he made a joke in the beginning about his knowledge of CDR sum of boxes, and I don't know that it was completely lost on the audience that he is the developer of that. So that is, he didn't name it after himself, so people don't necessarily associate it. But there is actually no one better to talk about how those scores break down than John. So
he did a great job.
This has been such a great discussion because I feel like we've really moved from the kind of diagnosis and the biomarkers all the way through the kind of basic biology back to the clinical trials and how we should be implementing them. So what a nice, well-rounded kind of overview of the drug development process. So on Friday, we had our plenary by Ken Marek, and he talked about a biologic definition and staging platform for neuronal synuclein disease. And then on Saturday, we had one by Pam McLean, so that was this morning, called Synergy in Science, Leveraging What We've Learned About Parkinson's Disease and Alzheimer's Disease to Inform Lewy Body Dementia Research and Therapeutic Development.
I've seen Dr. Ken Marek's presentation at a prior meeting, or a similar presentation at a prior meeting, and I do think it was, so much of what we've talked about even here is this idea is the underlying biology and how that defines or how that contributes to the progression, to the clinical disease across the board. And within, and Dr. Marek talked a little bit about this, that within Parkinson's disease, an individual clinically could have very different symptomology, but their biology is consistent or similar, and why, or at least the biology that we can measure today, and that we understand about today. And so how do we translate that to a diagnosis? And so this idea of neuronal synuclein disease, or NSD, it was published about a month and a half ago-ish in Lancet, and it really sets this idea of biological staging of the disease, a biological definition of the disease, and allows us to take what we know today, but then also sets a stage to move earlier as we think about therapeutic development, as we think about clinical trials. It's very similar to what is happening in the Alzheimer's disease space. It's also what the Huntington disease community has done. And so as we think about that trajectory of defining the biology, thinking about that as we think about treatment, as we think about diagnosis across the board, and getting to our conversation just a few minutes about comorbidities, that really you can start to move in that space where you can define and stage people based on where they have, and hopefully better understand on a personalized way what may be contributing to their clinical symptoms. So I thought Ken did a really fabulous job of defining and explaining the work group's work in setting out the paradigm of neuronal synuclein diseases. It was really clear and great response. It seemed to be a great response from the audience.
Yeah, it's nice to have tools to be able to do that, right? To define by a pathology like synuclein, right, where you now can try to measure and quantify, at least positively identify people with synuclein disease through different techniques. It's not something really we had before, right? We just had clinical diagnosis.
Like PET changed the game in Alzheimer's with our understanding of when those changes may have happened, and even further than with CSF and now plasma, we're doing the same with some seeding assays and Parkinson's.
I think it makes so much sense to have it based in the biology, especially with Parkinson's disease, where we always say that the area of the brain that's damaged determines the symptoms someone has, but the pathology defines the disease. And this is so important for something like Parkinson's disease because, I work in a big metabolic center. We get lots of mitochondrial disease patients. Almost, there are particular mutations in in nuclear mitochondrial genes that cause Parkinsonian features. They cause nigral degeneration. Is it the same as Parkinson's disease? Of course it's not, because it's got a different etiology.
We're almost out of time, but before we finish, I just have one more question for you guys quickly. So, from what you've learned over the last few days, what excited you the most, and what do you think is the next big discovery on the horizon? So just briefly, guys, so let's start with you.
Yeah, I have to say that I think the thing that I've been the most just in awe and excited by is the energy and the collaboration and the environment, the ecosystem that we're in. It was great to see so many students and postdocs presenting throughout the week, presenting their science, but also really thinking and challenging the ideas and the questions, and in bringing in new perspectives. You made the comment earlier about immunology and neuroscientists, but I think there's more than that. There's also individual, people that have vascular expertise, people that have metabolic expertise, and more, and that has really been represented all week with the discussion. So while I don't know that I would say any one particular thing, I think more generally around, I think what gives me a lot of hope is that ecosystem that we're living in, and really this multidisciplinary, cross-disciplinary, highly collaborative type of environment, and really that next generation of scientists that are clearly present in all corners, that we're seeing them really come forward with presenting their work and moving it forward.
It seems as promising as it feels when you put it like that. Okay, so Tim.
That's great. For me, I think it's, in terms of what the space has been like for so long, right? Now that we finally have our first FDA-approved therapy for Alzheimer's disease in particular, right? We can start thinking beyond that, right? With new ideas, new creativity, not just in how to make those therapies better by getting more antibody into the brain or removing it faster, but thinking about, orthogonal combinations in the future, keeping an open mind about, ways to target the disease that isn't just so focused on amyloid over and over and over again, and thinking more diverse. And now it's kind of starting to bring these groups together that maybe at one point were a little bit siloed and at odds with each other. So building that community across different types of pathologies or disease mechanisms is really exciting to see.
I like that, building the community across I like that.
Yeah, and I have to say I agree with both of you. I think that it's the collaborative atmosphere. It is people challenging things before, but I think it's also open-mindedness. I think people are much less siloed now. People are much more willing to listen to alternative viewpoints, and that can only be, of benefit to the whole field. Absolutely. So yeah, there is a great energy, and I think that's what we'll take going forward, I hope.
Well, I'm sorry to say that that's all we have time for today. And the conference is just so big that there's no way we could have covered everything. But I hope these two podcasts have given you a peek into what's been discussed, and that it's inspired you to check out some of the talks and the speakers online. So for now, I would like to thank our brilliant guests, Dr. Heather Snyder and Dr. Timothy Siegert and Dr. Daniel Erskine. So I'm Emily Maguire, and you've been listening to the Dementia Researcher podcast, live from the ADPD conference in Lisbon.
Thank you so much.
The Dementia Researcher podcast was brought to you by University College London with generous funding from the UK National Institute for Health Research, Alzheimer's Research UK, Alzheimer's Society, Alzheimer's Association, and Race Against Dementia. Please subscribe, leave us a review, and register on our website for full access to all our great resources. dementiaresearcher.nihr.ac.uk.